406. Probably because I spent almost like an hour when I was gonna share one. Like, what? I don't need to.
Yeah.
I'm not Gatorade. It's Hovig. You know Hovig?
Yeah. The exams are.
I know, and he's so yeah. Today.
How
Finish.
okay.
And you're just running all throughout the day.
At night, I always feel like
I feel like say that's like it was not
See.
Okay, so this is our last, at this point in the semester, our last lecture on nucleic acids. So we will be moving on to amino acids and proteins. I'll introduce that idea at the end, but so in finishing up speaking about nucleic acids, we wanted to spend some time looking at some experimental evidence for how DNA was replicated. So at home, you learned about the process by which the structure was elucidated, the methods by which they figured out it was double-stranded and had the sort of helical structure. Once that information was determined, it indicated potentially that the double-stranded DNA structure could have the ability to direct replication. Each one of its strands could direct replication of daughter strands. But the method by which it did that was not known. So this experiment details the models that were proposed and then the experiment that was done to show which model replication undergoes. So the three models that were proposed are called conservative, semi-conservative, and dispersive. And the concept here is that you have a double-stranded piece of DNA, and it directs replication of daughter strands. The way it does that could be one of three methods. One is that at the end of replication, you conserve the parental strands together as a base paired structure. And you have two daughter strands that are base paired to each other. Okay, that's called conservative because it conserves the parental strand as a unit. Semi-conservative is a model by which each parental strand is conserved in terms of its own structure, but the daughter strands are now base paired to one of the parents. So you get two double-stranded molecules that look like this. One parental strand exists in it, and then one daughter strand. And then the third potential model was something called dispersive, where the parental DNA and the daughter DNA was sort of intermingled, and you had pieces of parental in a linear sequence with new daughter strands and so on. So the parental DNA was dispersed within the new daughter DNA. And then each one of those was base paired to each other. Does anyone know which model is correct? Yeah, semi-conservative. Okay, so we know that, most of us. How do we know that? It is related to this experiment called the Mendelssohn-Stahl experiment. And this is, on our problem set today, we're going to be answering some questions to interpret data like this. So understanding the setup of this experiment is important. It's also a big MCAT type question to interpret this type of data. All right, so the setup, the way they did this experiment was to rely on the fact, again, radioactivity big, so we're using two different isotopes of nitrogen here, meaning they have different quantities of neutrons. So there is the presence of N15 or N14. Those are, we'll call them heavy and light nitrogen. And you can provide that in the context, so most of these experiments are done in bacteria. So we're looking for DNA replication in bacteria because that's easy to grow and look at. And the way that would work is in the nutrient media where the bacteria are replicating, you would either provide heavy isotopes of nitrogen or light isotopes. How does and nitrogen get incorporated into DNA? Like, where is it in DNA? Yeah, it's in the bases, right? So as bases are being synthesized in those cells, some of them will incorporate the heavy nitrogen versus the light nitrogen. Okay, so that's how that works. And so that's how the DNA is labeled. In this experiment, the purple will contain heavy nitrogen and the blue contains light. Okay, so fine, you're growing the DNA under these different conditions. You're allowing it to replicate under different conditions. And then how do you detect if there's either heavy or light nitrogen in the DNA? And that's defined here by its density. Heavy DNA has a density that is heavier than the light DNA. So the way that they determine this, how heavy is it, is to use what's called a density gradient. So the way you set this up is to have a solution of, in this case, they're using cesium chloride, but you can also do sucrose. Basically, you dissolve this complex, either cesium chloride or sucrose, in water, and you put it in a centrifuge tube. And then you put it into a centrifuge and you spin it really fast. And that solution, in that situation, because of the force of the centrifuge, more of the cesium chloride migrates towards the bottom of that tube and less stays at the top. And so it forms a gradient in that tube of cesium chloride. It's a higher density at the bottom, lighter at the top, and there's a gradient throughout the tube. So that's how you set up a density gradient. Now, you would take whatever sample you're trying to figure out what density it has. So in this case, if we took this solution of, if we took a sample of DNA from those bacterial cells and we layer it very carefully on the top of the centrifuge tube, you can take it out of the centrifuge for a while, layer your sample on top, you put it back into the centrifuge and you spin the tube again. The DNA is going to move through the tube until it migrates to the same density as whatever the cesium is at that location. So it migrates through until it sort of reaches a point where it's the exact same density as the cesium. And then you would take out the tube, you take samples of the tube, and of the various densities here, they are measuring if there's DNA there by absorbance at 260. So they're taking samples out of the bottom of the tube, usually like create a little hole, you take out samples, and for each density, you may or may not have absorbed. And that tells you if there's DNA at that density location. Okay, so that's the setup of the experiment. Any questions on that basic setup? Yeah.
So after it's put in the centrifuge, all goes to the density of the cesium chloride.
Yeah, so that you can imagine that in the tube there is, because you pre-set up the tube, there's going to be a lot more cesium down here and then less as you get up. So something like that, visual. The DNA has a certain density because of its composition of N15, but also everything else that's incorporated in nucleic acid. It will move through the tube until it is at the matching density to the cesium, wherever that is.
And you do this twice.
Well, the first is just to create the gradient to begin with.
Okay.
And then the second one is to migrate your sample through there. Yeah. Okay, so this is a look. These first two are just a look at the density for each of those molecules, whether they are grown in heavy nitrogen or light. And then the experiment is that we've started off with DNA that's been replicating in a heavy nitrogen condition. So all the DNA in these cells starts out at this density. Okay, so then the experiment is to take out that media and you exchange it for media that contains the light nitrogen. So the parental strands will look like this. Then we grow them for 1 generation, allow 1 round of replication with N14, the light nitrogen. Okay. So we have 2 new sets of daughter strands, and the absorbance is at this density. That's where the DNA, the resulting strands of DNA migrate to this density. Okay. Is this conservative replication? What would you expect to see if it was conservative replication? Separate peaks, right? So probably a peak here and then a separate peak there, right? That would make sense for conservative replication. You would have conserved parental strands as base paired together and 2 daughter strands grown in N14, which show a peak here. So it's not that. We have something in the middle. So what model does that support? Semi-conservative, because we know it's semi-conservative. Does it support dispersive? No. Why not? Yeah.
Then only have 1 peak at the middle.
Yeah. I mean, I think it doesn't discount dispersive, right? I think it could be one or the other. Because remember, dispersive is something like this. So I would expect to also see an intermediate peak if we had that type of replication. Okay, so then they've eliminated 1 possibility. So then they allow another round of replication to continue. Again, we're still only in the light nitrogen. So all potential daughter strands are going to be N14 light density. And then they get this result. Okay, so each one of these strands, 1, 2, 3, 4 strands, they come apart. They each direct a daughter strand to be replicated, but all daughter strands, remember, have to be light nitrogen. That's the only nitrogen that's present in the cellular condition. So now they get a result where there is an intermediate peak and a light density peak. So what model does that support? Semi-conservative. Is there a possibility it's dispersive or no? Probably not, right? So that sort of eliminates the dispersive replication. What do you think you would see if it was dispersive? Probably 1 peak. It might be getting like less dense because overall there's less heavy nitrogen around, but you could still kind of expect 1 peak. It might be not right here anymore. It might be slightly lower or something like that. But it would still, the parental material would still be dispersed throughout. So yeah, you would still expect 1 peak. So that sort of second round of replication allowed them to narrow it down to predicting semi-conservative. Questions on that experiment? Yeah.
So if we're getting like 2 in the middle, like those like 2 blue ones, how is that semi-conservative if they're both like the same?
They are, and that is because you have, this is a parental strand now. For the second round of replication, this blue is a parental strand and its daughter is also blue. Remember, the only potential daughter material is light nitrogen. We're not switching back and forth between heavy and light. So the only heavy DNA is from the original starting condition. So each one of these strands comes apart, and now we have each one of those as a parent. So parent, 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 and parent. We have 2 blue parents and 2 purple at that point. Questions on that? Any other questions? Okay, so we'll do some interpretation of more experiments like that in our problem set. All right. So looking at secondary structure of nucleic acids, we'll hear these terms a lot as we go through, especially in proteins. We talk a lot about primary, secondary, tertiary, and so on. there is a little less focus on tertiary structure in DNA, because they're very large molecules. So looking at a three-dimensional structure of a whole piece of DNA is not easy. So we really mostly focus on primary structure. Primary structure is just the linear sequence of nucleotides. Secondary structure is what it folds up to in a local location. But for DNA, it tends to follow that structure throughout its whole molecule. So what we think of as double-stranded DNA really is it's called B-DNA, and it is a right-handed helix. So it's right-handed because as the DNA twists, and if the direction of the twist is towards our thumb, it is twisting in a right-handed way, going up, not in a left-handed way. So it's a right-handed helix. There's about 10.5 base pairs per turn. And then the rise, you don't have to memorize these numbers. The rise is about 0.34. So if you're thinking about like a stairway, the distance between stairs is 0.34 nanometers. And then the bases are base paired along the interconnection here between the two strands. We have the hydrogen bonding across there. And then like we saw in the at-home part of the lecture, when you look at the space filling model of DNA, here, how they're sort of atoms occupying space, they really stack right on top of each other. So the DNA is held together with hydrogen bonds, but when it is bonded, you start to get the van der Waals interactions because the bases are so closely stacked. Okay. And then this is a look at top down, how you have the base pairing across the structure. In other forms of DNA, or depending on if you have a double-stranded DNA in a low humidity environment, meaning it's probably an experimental condition where you have not a lot of water around, it will adopt a right-handed aDNA structure. And you can see it's a little more squat, slightly more base pairs per turn, and the rise is a little less between successive nucleotides. So that's called an aDNA structure. So that's DNA in a low humidity environment, but it's also double-stranded structures of double-stranded RNA forms that, or DNA RNA hybrids form that. So those are other types of secondary structure of nucleic acid. And you can see top down what the base pairing looks like there. It's a little different than straight across the strand. Okay, tertiary structure. This is sort of what the whole molecule looks like in three-dimensional space. DNA is really large, so we usually don't have a great descriptor like we do tertiary structure of some proteins. But it can exist in, it can be wound around histones, right? And then it's very organized by proteins. If it is circular, so human mitochondrial DNA is circular, E. coli chromosomes are circular. In this case, we can really see that it is relaxed. It's sort of an open circle, but it can also be folded over on itself in this supercoiled structure. And that is useful for compacting the DNA into a nucleus. That's supercoiled DNA. This is a relaxed circular DNA. And this is sort of what it looks like in cartoon form. So this would be a double-stranded, relaxed circular piece of DNA. And this is supercoiling. It's the whole molecule flipped over on itself. And you can coil it in one direction or in the other, and that indicates a positive or a negative coiling. You don't need to worry about these numbers now. You'll talk more about supercoiling and so on as it relates to DNA replication when we get into that section with Dr. Castro. This is just some examples of the tertiary structure of DNA. Okay, we can also have secondary structure of nucleic acids that are single-stranded. And these are some examples. So typically single-stranded nucleic acids are going to be RNA. That's usually what we see in a cell that is single-stranded. So it can be a random coil like this where there's no really defined structure. It could be in this form where the bases are stacked on top of each other and it forms sort of a turned structure, or it could be quite defined in terms of its secondary structure here, where there is intramolecular base pairing between the single-stranded molecule and itself. So it has some sequence that is self-complementary and forms base pairs. So these are called stems or stem loop. If it looks like this, where this is a single RNA or a single-stranded piece of DNA, part of the sequence is complementary to itself, and so it forms base pairs across that structure. The loop occurs because this part of the structure does not have self-complementary regions. So it forms this sort of unbase paired loop and then base pairing there. So that's called a stem loop. We see this a lot in tRNA molecules, right? Highly structured RNA involved in translating an mRNA codon sequence into proteins. This has 3 stem loops and a stem here. The anticodon, this end is what sits down on mRNA. The other end brings in the amino acid. And so questions have been asked, are these stem loops important biologically? Can you mutate them and remove this base pairing? Will it still function to translate? And the answer is no. A lot of it depends on how extensive the mutation is, but if you remove the structural components here, these stem loops, then it doesn't function as a tRNA. So they're very important biologically. We see a lot of things like stem loops in RNA viruses. So quite often the ends of RNA viruses have stem loops. It's important for the virus for replication, for packaging up the viral genome into the virion. So these structures are quite important biologically. Okay. So as any questions on the, yeah.
When they, like the single strand nucleotide base pairings kind of like go together and create a loop, are they still hydrogen bonded between the pairs?
Yes, exactly. So it's the same. There. Yes, same hydrogen bonding that we expect in a double-stranded DNA structure is occurring here. Yeah, so you get the same look at it. 3 hydrogen bonds between GCs and 2 between AUs, just like an AT has 2. Yeah.
Just to confirm, the stem loop is the longer structure, or is it the circle?
So this is called the stem, the base paired portion, and the loop is the unbase paired portion.
Okay.
Yeah. Anything else? Yeah.
What is in the tRNA molecule? What are the X's for?
Good question. So those are areas where the nucleotide is not as important. So yeah, you can have some variability there. So when, so he's asking about these X's, and that means that those can have some variability in which nucleotide is in the tRNA of those locations. Good questions. All right, so here I will want to talk a little bit about the structure of DNA and experimentally how we think about that stability of that structure, the double-stranded nature. So what we know is that this is what we call the native form of DNA. It's double-stranded when it's complementary But what happens when that is heated up is that it denatures, it comes apart into two single-stranded structures. We call that denaturing, we call it melting. Those terms are both sort of used interchangeably. So how does it go back and forth between this? How do we detect this? If you have a piece of DNA, so we know that at 260 nanometers spectrophotometer, we can measure concentration, right? Talked about that the other day. So if we look at absorbance at 260 nanometers, and we have a certain absorbance based on how much DNA is there, what's interesting is that if you increase the temperature of that sample, at a certain point, the absorbance will increase when it becomes denatured. What is it about a denatured structure that absorbs more light than a native structure, even though you're not changing the concentration? What do you think? Why does this absorb more light than that?
Is it the exposed base pairs?
So the exposed bases are absorbing more light. In the double-stranded structure, the bases are what absorb light. And when it's in a double-stranded structure, the bases are somewhat hidden by the phosphate backbone. So they absorb some amount of light, but when you denature them, they're more exposed. So they absorb more light, even at the same concentration. So this absorbs more light. And we can detect that for any piece of DNA, it has a so-called melting temperature. And this is, the melting temperature is the temperature at which, essentially this point, where 50% of it is melted and 50% of it is heat. So that's called its melting temperature. So you can calculate this. If you have a piece of DNA that's 100 base pairs long, its melting temperature, first of all, depends on its length. If it's 50 versus 100, which one is going to have the higher melting temperature? 100. It takes more heat to melt something because we have to break those hydrogen bonds. Other things that affect melting temperature are the relative quantity of GC base pairs versus AT base pairs. GC base pairs have 3 hydrogen bonds, AT have 2. So melting temperatures will increase if you have more GC base pairs. Okay. So, and that's up to a certain point. Once you start talking about like genome level length, at that point, the length doesn't matter as much. It's just going to melt at a certain point. But when you have shorter pieces of DNA, the melting temperature can vary quite a bit based on its length and GC content. A lot of people calculate the melting temperature, especially when they're setting up experiments like PCR, and they're asking, what temperature does my PCR primer come off of the template? Okay, so we know that heat causes this to melt and denature, and we can think about the tendency to adopt one or the other of these conformations based on delta G. So if we raise the temperature, the delta G is negative in what direction? If we raise the temperature, delta G is negative towards denaturing or being full. Yeah, denaturing, right? It's favorable to come apart when we raise the temperature. It is favorable to be double-stranded when the temperature is lower, right? They base pair back together. So if we think about this in terms of the delta G, it starts to make sense because in which direction do we increase entropy? The denatured condition, right? That's a more random structure, has more random conformations. So the entropy increases in this direction. And so if you increase the temperature, the gain in entropy in that direction has more impact on the delta G being negative in that direction. The delta H is negative in this direction when you form the base pairs, the hydrogen bond. Okay, and so which one has more of an impact on the delta G in which direction is going to depend on temperature. So high delta T means that the gain in entropy in this direction overpowers the drive towards the hydrogen bonding in the opposite direction. Questions on that? Yeah.
So that 260 nanometers, we can measure the concentration of DNA.
Yeah, DNA or RNA. Yeah, any nucleic acid concentration can be measured at 260.
Thank you.
Yeah. Okay. Leaving nucleic acids here, moving on to proteins, you'll see that the at-home lecture for Monday is a little longer than normal. And that's because talking about individual amino acids is boring. So a lot of that is at home. It's just going over the amino acids, their characteristics, and so on. We will still talk about proteins in class, but to get started, you can start making your amino acid flashcards. So the goal would be you can draw out all the amino acids. You're not going to have to draw them on the test, but you have to know enough about their structure so that you know about their chemistry. So if I ask you, what does glutamic acid do at pH 2? What does it look like? Like what's the charge on its amino end chain? If you don't know what it looks like, you're not going to understand that. So you don't have to memorize every atom, but have the goal of being able to draw them. And if you're just somewhere in between and can sort of visualize it, that's great. Other things you can start learning are the 3-letter codes and the 1-letter codes for the amino acids. Those are things you will also come upon on the MCAT. So learning them now is beneficial in both ways. Yeah, so I just wanted to sort of alert you that that is upcoming so that you can start getting on top of that. Because the quicker that you learn those, then the easier it is to think about the types of interactions that those amino acids have in a protein structure. Okay. All right, so let's do the problem set. We still have plenty of time to get that all done.